Today we're going to discuss the last section of Chapter 4, how proteins are controlled. But that's going to be our main topic. But first, I'd like to just finish up how proteins work from the previous section. So I'm going to add a page here and just discuss a couple of other points that have to do with both proteins in general and enzymes. And it's that sometimes some proteins, some enzymes require other small molecules bound to them to do their jobs. It kind of extends their functions, gives them some functionality that proteins don't have. And an example of this is a molecule we looked at in class, hemoglobin, the molecule that, the protein that carries oxygen around the blood. And we saw in class that it's a tetramer composed of 4 different polypeptide chains, and each of those chains had a non-covalently bond heme group attached to it, and that heme group had the iron inside of it that binds oxygen and helps hemoglobin carry oxygen around the blood. Many enzymes also have bound small molecules that help their function, and they fall into 2 classes of bound molecules. There are cofactors, and these are inorganic bound small molecules or ions that help often catalyze the reaction in enzymes. So, for example, zinc can be in the active site, helping to form the product. Also, there are coenzymes, and these are organic small molecules that help the enzyme function. And one of these is often biotin that helps transfer carboxyl groups. And biotin, there's several forms that come from different forms of vitamin B, and these help enzymes that we'll look at when we look at transfer of energy in cellular respiration. So again, these small molecules can be necessary for an enzyme to carry out its functions. And again, the cofactors are inorganic and the coenzymes are organic. So now we're going to look at how proteins in a cell are controlled, at least some of the major themes. Protein activities are controlled at many levels. On the grandest scheme, the cell can regulate gene expression or synthesis of proteins, so that specific cells may or may not have one of the proteins or many of the proteins expressed that could be expressed from the genome. So that's one way of getting the proteins into the cells where they're needed. Another thing is that there are still many, many hundreds of proteins active in every cell, and compartmentalization helps keep certain proteins and activities to certain regions of the cell, especially eukaryotic cells where we have different organelles carrying out different types of reactions. But what we're going to look at in the rest of this chapter is really how protein activity can be directly regulated. So there's a list of ways that it can be regulated below here. And we're just going to go through these one by one and look at some examples of each of these types of regulation. So we're first going to look at positive and negative regulation of proteins. Just as their name implies, positive regulation increases the activity of a protein, whereas negative regulation decreases it. And a common way to control enzymes this way is that molecules that aren't the substrate bind to the enzyme at either the active site or a separate regulatory site. So this figure shows that in feedback inhibition, it regulates further progress or inhibits further progress toward the product of the reaction. So, for example, in this figure, B here is the reactant for the final product Z, and the end product Z, this is a common type of inhibition called feedback inhibition, and Z would be the feedback inhibitor. This product is feeds back and inhibits the first enzyme in the pathway. So thereby it limits its own concentration in the cell. And again, this type of negative inhibition is called, or regulation is called feedback inhibition. But you can also have positive regulation, and that's shown in this figure as well. If there's a lot of this reactant around A, it can regulate the flow through the pathway as well. So in this case, if there's a lot of reactant, there's probably less of the product, and this reactant can feed forward, it's called, and activate this first enzyme in the pathway, thereby causing the reaction to move forward faster in the making of product Z to use up A. So this is very much, I think of this like supply and demand in economics. This, if you don't have enough supply, we're going to stimulate making more. If there's too much, we're going to inhibit making more. So as the chapter 3, that section on enzymes, that's kind of in the middle of the chapter, I was having you look at that because it talked about enzyme activity and how it could be measured and the types of inhibitors that affect enzymes. So I wanted to look at inhibition for a moment. And one way that enzymes can be inhibited is through competitive inhibition. And so this diagram is showing an example of this. It's showing an enzyme here and the enzyme's active site. And normally the substrate would bind to that active site and the reaction would proceed. But there are inhibitors called competitive inhibitors that bind directly to the active site, and they block the binding of the substrate, as they show over on the right here. So that as long as that inhibitor is there, that enzyme is not able to function. So in lab this week, we were looking at enzyme kinetics, where we did an enzyme reaction at increasing concentrations of substrate. So ultimately, you'll be making graphs where you're looking at the rate of enzyme conversion of substrate into product, and looking at that over a range of increasing substrate concentrations, and you'll get a plot called the Michaelis-Menten plot, where the speed of the reaction changes over time, ultimately very fast, and slows down as the enzyme becomes more saturated. Well, in the presence of a competitive inhibitor, the kinetics change, and you can measure that in the same type of experiment we did, where you do the enzyme reaction in the presence of the competitor. So let's see if I can get a different color line. Here we go. And with competitive inhibition, you get something that looks like this, where ultimately the enzyme can reach the same Vmax if there's enough substrate added to kind of outcompete the inhibitor. So you can reach that kind of same level, but if you notice here, let's see, I might use another color, the Vmax of the enzyme, like the halfway point, let's say it's here for this one and here for this one, so, half maximal speed. Okay, so that gives you a value of the Km, or the Michaelis constant, how much affinity the enzyme has for its substrate, and that does decrease. So with competitive inhibition, in kinetics, you see the same Vmax, but a different Km. The other type of inhibition is non-competitive inhibition. And in this case, nothing is competing with the substrate for the active site, but there's another molecule, a non-competitive inhibitor, that will bind to a different site on the enzyme. It's sometimes called the allosteric site or the regulatory site. And this binding of that other molecule prevents the binding of the substrate because it changes the conformation of the substrate so that it can't interact with, of the enzyme, so that it can't interact with the substrate. And this, in terms of enzyme kinetics, this gives us a different type of curve where if we look at velocity again, the rate of enzyme reaction against substrate concentration, if this is our enzyme without inhibitor, then the curve with the inhibitor looks like this. So here the Vmax is different between the enzyme with and without inhibitor. So Vmax is different because none of the enzyme can ever be fully bound, because if there's inhibitor there, some of it's going to be in that non-active conformation, and the substrate can't compete with that because it's binding to a different site. But if you look at the half maximal rate, where you can figure out the affinity of the enzyme for the reaction, the half maximal rate of each of these, I didn't draw it perfectly, but the Km should be the same. So often the Km is the same for this type of inhibitor, and the Vmax is different. Again, that's the opposite of what we see in competitive inhibition. So here is an enzyme, a model of it, that is regulated by an allosteric inhibitor. And the inhibitor, well, this enzyme is aspartate transcarbamylase, and it's an enzyme from E. coli that begins the synthesis of pyrimidine rings for DNA or RNA, and it is inhibited by one of the pyrimidines, cytosine triphosphate. So if there's plenty of one of the products around that it's going to be making, it's inhibited. And again, this inhibition doesn't occur at the active site, which is in between these two subunits. So there's a lot of different units, subunits to this enzyme. It's quite complicated, but The active sites are shown kind of in red, and the sites where the CTP binds, there's 4 different sites, but they're outside of the active sites, and just change the conformation and make it inaccessible to the substrate. Now here's an example of feed-forward activation by an allosteric activator. This is kind of a hypothetical example, but they're showing an inactive enzyme here on the left and an active enzyme down at the bottom. And what's activating the enzyme is ADP. So this is an enzyme that would break down, help, in the pathway to break down sugars and get energy. And so if ATP is low and there's a lot of ADP in the cell, it will stimulate that enzyme to work. And what these regulatory molecules do is they really affect the equilibrium between the active and inactive conformations. So they're showing, say, without ADP, only about 10% of the enzyme is in the active conformation, whereas with ADP, it's facilitated the change to that active conformation, and now much more of the enzyme is active. And then the allosteric inhibitors would just inversely affect that equilibrium. The last thing I'd like to point out before moving on to another type of regulation is that inhibitors of enzyme activity can be irreversible, in that, for example, a competitive inhibitor combined in the active site of the enzyme covalently and not come off as the other inhibitors do that are just controlling metabolism. And this is a common mechanism for nerve gases and other toxins, poisons, antibiotics, other drugs. So in this example, they're showing the enzyme acetylcholinesterase, which is important for breaking down the neurotransmitter acetylcholine that transmits signals from nerves to muscles, among other things. And so there's this inhibitor, DIPF, which in this case it's a nerve gas. The full name I won't bother trying to pronounce at this point, but it binds to a serine residue in the active site of acetylcholinesterase and permanently deactivates it, so that acetylcholine is not broken down in the synaptic cleft between the nerves and the muscles. And this eventually leads to overactivity of these synapses, and then the ability not to be able to transmit impulses at all. So this is a nerve gas, but antibiotics, many of them work in similar ways, where they're inhibiting specific enzymes in bacteria that carry out functions necessary for their existence. So this is a mechanism, again, that is how many drugs and toxins work. The second type of direct protein regulation is protein phosphorylation, and this is a covalent modification of where one or more phosphate groups are added to the protein, and these groups are usually added from one of the high energy carriers, such as ATP often, or sometimes GTP. So we're going to look at an example of each. And this phosphorylation controls the activity of many proteins in eukaryotic cells. So here's that word, phosphorylation. I think we've talked about it before, but again, it's just adding a phosphate group to a protein. Covalently, usually attaches it to the hydroxyl group of a serine residue, like in this example, this is serine, or threonine or tyrosine residue in a protein. But this phosphate group changes the conformation of the protein, as you've got these kind of negatively charged groups coming in, with causing that conformational change. And as I said, it controls the activity of many proteins, and scientists have measured like over a third of the proteins in mammalian cells are phosphorylated at any one time. And this phosphorylation usually occurs in response to signals from either inside or outside of the cell that change the state of the cell. Like protein phosphorylation happens all over the cell during cell division. So there are hundreds of the enzymes that are involved in phosphorylation in cells. This example on the left, A, here, again, they're showing the protein and the serine side chain that's modified by phosphorylation, and the enzyme, or a whole group of enzymes that does this, they're called protein kinases. So a kinase is any enzyme that's going to add a phosphate group to a protein. And these can be removed, so this is a reversible process. And another group of enzymes removes the phosphate group, and these are called protein phosphatases. So we'll be talking about these later and looking at how they work together to regulate protein activity, especially enzyme activity. Now, the other example here on the right is showing how GTP can control the activity of a protein. Oh, actually, this picture on the right isn't for GTP. It's the same type of diagram they used in that one, but sorry, this is showing just in a simpler form how phosphorylation can switch on or off protein activity depending on the protein and the pathway it's in. So up at the top, they're showing here phosphorylation is switching on a protein, and here it's switching off the protein. But they do act as kind of molecular on-off switches. ATP binding to a protein can also lead to some interesting properties in the activation of that protein, as the ATP can be hydrolyzed while it's on the protein, and the binding and release of the ATP or the ADP and phosphate activate different it kind of caused different conformational changes. And this is a great example of this is motor proteins. So this green kind of half of a stick person represents a motor protein. And these proteins generate the forces that cause muscle movement, and also most every other kind of movement, biological movement. They move cilia, flagella of eukaryotic cells. They cause the kind of crawling movement of amoeba. They move organelles inside of cells. So these motor proteins are really essential. And the way that ATP binds and powers them allows their movement to be directional. So they can move from point A to point B, or move something that's attached to them from point A to point B. So the way that they can move forward or in a particular direction is that because they have ATP molecule directly bound to the protein, when it's hydrolyzed or breaks down into ADP plus a phosphate, that's a huge amount of free energy is released, and it makes it unlikely that that protein would ever go back to the reverse shape. It's going to go forward to the next shape, moving in one direction. So many motor proteins generate this movement by using hydrolysis of tightly bound ATP, but we'll see later that also there are molecules that proteins that also bind GTP, and that hydrolysis is really important in signaling systems within the cell. So we already talked about protein phosphorylation, and as I said, that was a covalent modification of the protein. But here we have another section on covalent modifications beyond phosphorylation that are also really important in the cell. In recent years, scientists have discovered there's more than like 100 different types of covalent modifications. And some of the common modifications are shown on this example. This is a p53 protein, which controls how a cell responds to DNA damage and other stresses. And it's often called a tumor suppressor protein. But as you can see, there's multiple modifications that can be on this protein, and they can all have different roles in its activity and function. But so they're showing that it can be phosphorylated, lots of sites for phosphorylation. It can be acetylated, which is acetyl groups can be added to the protein. Ubiquitin, which is a group, a chemical group that kind of marks the protein for degradation. There's sites for that on this protein. So there's a lot of different ways to regulate the protein, and I think the more central it is to the survival of the cell, the more closely and tightly it will be controlled. So a lot of proteins work together to form complexes that do really complex things in the cell, such as DNA replication, gene transcription, synthesizing proteins, signaling across the membrane. So we call these big complexes of proteins and enzymes working together machines often. They're big protein machines. And they work together by binding ATP or GTP, as we were talking about earlier. And when that ATP hydrolyzes or the GTP hydrolyzes, it causes conformational change that allows these proteins to interact together to perform a specific function for the cell. So they give an example here, kind of a make-believe example, but of a group of proteins on the left that's going to work together to turn a key and open a door, say, on a safe. Maybe you could think of this as working together to replicate, a side of a DNA molecule. So in this example, each protein undergoes conformational changes when the ATP bound to it is hydrolyzed. And if they occur in this large protein assembly in kind of a coordinated fashion, they'll cause these proteins to work together again to perform a specific function. And then at the end, when the ATP and GTP or ADP and or GDP and phosphate come off, they go back to their original conformations, ready to bind more ATP. So this is an example of how proteins can work together using phosphorylation as an activating factor in this cooperation. So we're going to see lots of examples of this, specifically as we start looking at, I think first we'll see it in DNA replication. The last section of the chapter looks at how proteins are studied, and we're going to talk about this in lab next week and as we go on throughout these chapters. So I'm going to leave this section for lab, but you might it might be useful to read through this before lab next week, especially the section, let's see, like pages 164 through 167. So that goes into some specific methods and how they work. And we're going to be using gel electrophoresis this week to look at proteins. And the last is just a summary of some of the main points. And then you also have your chapter study guide for this chapter.